Okay, so in this video we will discuss on the range of reproduction in Chlorophyceae. Now in the previous video we had studied the different structures, vegetative structures in green algae. So in this video we are going to focus on reproduction. So as you have already studied about the life cycle of Chara and Idogonium, you have seen that they usually reproduce by asexual, vegetative and sexual methods. Okay, so reproduction in green algae in Chlorophyceae, it takes place by vegetative, asexual and sexual means. Now coming to the first one, okay, the vegetative reproduction. Now here, the propagation of the species by any method which uses only the vegetative cells is known as vegetative reproduction. And in this method, the parent cell wall is retained. Okay, and it usually takes place by several means. So let us take a look at these different types or several means by which the vegetative reproduction occur in Chlorophyceae. Now the first one, as you can see here, it is through fragmentation. Alright, so what happened in fragmentation? Here it involves the breaking up of multicellular filamentous thallus into one or many cell fragment. As you can see here in this figure, in case of Spirogyra, you can see here this filamentous thallus, it breaks into two or more or many cell fragments. You can see here it breaks into many fragments, and the fragmentation of the filament, it may be the result of the external forces or the formation of mitospores or gametes, okay, in certain parts of the filament, followed by the breaking up of empty cells. Now the fragments, or by repeated cell division, they will then grow into new filament. Okay, so as in this case, you can see in the case of Spirogyra. In Spirogyra, not only in Spirogyra, we had studied even in Idogonium as well as in this fragmentation, it occurs even in Eulothrix, okay, Eulothrix species. Now the second type is the acinids, okay, so vegetative reproduction through acinids. Now again, this structure you have seen, we had talked about this in case of Idogonium. So Idogonium, you know that it is Chlorophyceae, but not only in Idogonium, in many other Chlorophyceae, such as Eulothrix, again, they usually form these vegetative structures known as the acinids. Now these are one cell, specially modified resting vegetative structures. Okay, they are the resting vegetative structures, and on the certain conditions, what happened actually, some cells of the filament, okay, this is the whole filament, but some of the cells under certain conditions, they will lose water and contract to become round or oval. And here the vegetative cells, they develop into spore-like stages, okay, with much thicker walls and more food materials, and they occur either singly or maybe in chains, okay, like this, in chains, or either singly. As you can see, this is not acinids, but this, it occurs singly in the vegetative filament. And these acinids, they can survive the unfavorable conditions, okay, the unfavorable conditions, suppose, because since they are referred to as the resting vegetative structures, okay, they can survive the unfavorable conditions, and when the favorable condition occur, then the growth will start to occur. So this can be usually this has been reported from several Chlorophyceae, okay, for example, like in Idogonium, Eulothrix and many other green algae. Now coming to the third type, here you can see it is through cell division. Okay, vegetative reproduction through cell division, also known as fission. Now here what happened in fission or cell division, the unicellular forms of the thallus. Okay, remember that we have in vegetative structures, we also have species which occur as unicellular cells. Okay, so in case of these species, they commonly reproduce by a simple process of cell division called fission. Now here, as you can see, what happened, the nucleus will divide. Okay, and not only the nucleus but followed by the cleavage of the cytoplasm, which begins by the median constriction. You can see here in the middle there is a constriction at the periphery, and the inner layer of the wall along with the wall membrane invaginate and the cytoplasm is then separated into two halves, and later on the daughter cells separate. Now in unicellular form, the cell division is a method of reproduction which in multicellular form it will lead to growth. Okay, for example, in case of Protococcus. In Protococcus, the cell division will lead to growth. But in case of multicellular form. But here, since in unicellular form, this is actually a method of reproduction. They reproduce by this method known as cell division. Now, coming to the next type, this one you are familiar with this one since we already studied the life cycle of Chara. Remember, the vegetative reproduction, it usually occurs by these what we call the Amylam stars. Okay, these are the multicellular aggregation of cells looking like stars. That is why the name Amylam stars, and also they are filled with amylum starch. Thus they are called Amylam stars, and such structures they are found in Chara, which help in vegetative reproduction. And also reproduction can also occur by tubers. Okay, tubers just like bulbils only, they are found in Chara, and such bodies they are full of starch. Okay, so by tubers also vegetative reproduction will occur. These will give rise to a whole new plant. They will grow into a whole new plant body. Now we have also we also have the secondary Protonema. Okay, secondary Protonema, you can see these secondary Protonema, they are thread-like structures which develop from these primary Protonema. Okay, from the basal cell of the rhizoid. And such structures again they are found in Chara. This is the primary Protonema and these are the secondary Protonema. The secondary Protonema may arise from the primary or from the rhizoid. They are thread-like structures which are found in Chara, which help in vegetative reproduction. Now the next type is bulbils. Again here you can see these are known as the amorphous bulbils, stem bulbils, or the root bulbils. So these are nothing but they are small oval spherical bodies, okay, which develop and aggregated at the nodes of the plant body. Again you can find this in Chara. And after detachment from the plant body, after they get detached, they will fall on the ground, germinate, and they will develop into whole new plant. So this type of structures also can be found in Chara. Now that is all about the vegetative reproduction. So coming to the next type, we have the asexual reproduction, and this type of reproduction is a common feature again in most of the members of Chlorophyceae. Okay, and it also takes place in several ways. Now the first one here, it takes place by zoospores. So the common method by which the asexual reproduction takes place in green algae is by the formation of these motile naked asexual spores called the zoospores. And these zoospores, they may be biflagellate or quadriflagellate. Okay, 2 flagella are present, or 4, or many. As you can see, in case this is the figure, if you remember, it is from Isogamy species. You can see the zoospore is neither biflagellate or quadriflagellate, but the zoospore, it is multiflagellate. Many flagella are present. Okay, and the protoplast of the cell, it may develop into a single zoospore, or it may, as in case of Idogonium, or in other species, it may undergo division resulting in the formation of many zoospores. Okay, not a single zoospore. And each zoospore is a tiny naked mass of protoplast which consists of single nucleus, an eye spot, or one or more chloroplasts. And those flagella, okay, in case of those species where multi-zoospores are formed, the flagella also, as in this case also, you can see they are of equal length. The next type we have here is through the second type of spores termed as planospores. Now these planospores, they are non-motile. That means they lack flagella. They are the asexual spores, often termed as planospores, and they are normally formed in algae of subaerial habitats. Okay, so those algae which are found in subaerial region, and they are sometimes referred to as the abortive zoospores. Okay, so the protoplast of the cell, it may form only one, okay, which is known as microspore, or as in the case of microspora, only one planospore is found, okay, is formed. Or in case of other green algae, many or more than one, as you can see here in this figure, more than one planospores are formed. And each of the planospore, you can see something which surrounds it. This is nothing but each planospore, it secretes a wall around itself, okay, before liberation from the parent cell. And also it lacks flagella, and thus it is non-motile. Now when these spores which are produced, okay, they appear identical to the parent cell, then they are referred to as the autospores, and they are usually found in colonies known as the autocolonies. So I repeat, when the spores which are formed, they are similar to the parent cell, they are referred to as the autospores, and they usually are found in colonies, and that colony is termed as autocolonies. But under certain circumstances, these planospores, they secrete thick wall around them, okay, and they store abundant food reserve. Now these thick-walled planospores, they are termed as hypnospores. Okay, these hypnospores, they are usually perinating bodies. Okay, they undergo perination. They are perinating bodies, and they are usually again found in many green algae. For example, in pedestrium species. Now coming to the next type of asexual reproduction is through this stage. If you remember again, when we discussed about the vegetative structure, I had mentioned that this palmilla stage is permanent in some algae, but in some algae it is permanent, such as palmilla. Okay, so this is, in case of palmilla, this is the vegetative structure that can be seen in palmilla. But this stage in some other algae, such as Chlamydomonas, it occur only temporarily. Alright, so in this stage, the individuals, they will divide and redivide till innumerable daughter cells are developed in a gelatinous matrix. Okay, these daughter cells, they are developed in a gelatinous matrix, and on the approach of favorable condition, this matrix will then dissolve. Okay, will then dissolve, allowing the cells to develop into new individuals. Okay, so under unfavorable condition, these will form. Right, they will form amorphous colony, as you can see here, with the indefinite number of spores, and is called palmella stage. But when the favorable condition it approaches, so the matrix, that gelatinous matrix or envelope, it will dissolve, allowing the spores to develop into new individuals. So this gelatinous envelope, it acts as a protection, okay, during unfavorable condition, and it is usually found in Palmella and Chlamydomonas. Now the next type here we have true daughter colonies. Remember again, this one we had discussed in Volvox. What happened? The daughter colonies, sometimes even the granddaughter colonies, they are developed asexually within the parent colony. Okay, when the parent colony it breaks the newly formed colonies, they will swim away and they will start forming new colonies. Okay, so this is usually seen in case of volvox. Next we have the last type of reproduction that we see in Chlorophyceae is sexual reproduction, and this kind of reproduction, it involves the fusion of two specialized reproductive cells or units called the gametes. Okay, now the fusing gametes, they may be produced by the same thallus, that means monoecious species, or by two separate thalli, that is in case of dioecious species. And sexual reproduction, it always entails 3 steps. Alright, that is the formation of gametes, then those gametes will fuse in fertilization, and the last step is germination of the zygote. And it takes place only once during the growing season, when the climax of vegetative activity has just been passed. And this again, it can be of 4 different kinds. Okay, that is isogamy, anisogamy, ugami, and one more we will see later on, that is a Plano Gammy. So in isogamy, this is the simplest and primitive type of the sexual reproduction. Now, as you can see here in the first figure, that the fusing gametes, they are alike in size as well as in their structure and also behavior. Such gametes, they are termed as the isogametes. Okay, and the sexual reproduction involving them is termed as isogamous. And these isogametes, they are usually naked protoplasts and are often flagellated. You can see the presence of flagella here. So these types of gametes, they are produced in ordinary cells known as gametanja. Okay, so they are produced inside the structures, that is the female and the male structure, termed as gametanja. And usually such gametes, they come from 2 different individuals, that is from male and female. One will come from male, one will come from female. And this type of reproduction is usually found in many Chlorophyceae, example or species of Chlamydomonas as well as Eulothrix. So coming to the second type here, that is anisogamy. In anisogamy, what do you see here in this figure? The gametes taking part in fusion, they are not at all identical. Okay, the gametes, of course, they are flagellated, and one gamete is larger than the other. The smaller one is considered as the male gamete, while the larger and less active one is referred to as the female gamete. And this example of reproduction, anisogamy, is again found in several Chlorophyceae, in Chlamydomonas brunei. And here in other species such as Spirogyra, both the gametes are similar and non-motile. Okay, they are similar, they are non-motile. The male gametes, it will move towards the female gamete through physiological anisogamy. So that is termed as physiological anisogamy. Physiological anisogamy, just remember, when the gametes, they are similar and they lack flagella. Okay, both they lack flagella, but the male, it will move towards the female by amoebic movement. And we have Ugami. This Ugami, it is the highest evolved type. In Ugami, there is a union of small flagellated antherozoid with a large non-motile egg. You can see why it is non-motile, because it lacks flagella, and the female gamete is usually large without flagella and is not shed. Okay, if you remember when we studied the different life cycles of green algae such as Idogonium, Cara, you see that the female gamete is not shed, it remains within the gametangia. Okay, whereas the male gamete, it is small, it has flagella, it is motile and is shed in the surrounding water. After that, the male gamete will swim towards the female gamete, it will become attached to it, and when it become attached through the intervening walls at the point of contact will dissolve, and the male gamete will come in contact with the egg or the female gamete, and it will fuses with the female protoplast, forming the zygote. And this can be seen in Chlorophyceae such as Volvox, Idogonium, Vaugeria and several other Chlorophyceae. And here in Ugami, the species, they may be homothallic, okay, monoecious. That means the male and the female sex organs which produce these gametes, they are present on the same plant. Or it may be, the species may be heterothallic or dioecious. That means the male and the female sex organs, they develop on two different thalli. Okay, so coming to the last type of sexual reproduction is a Plano Gammy, which is not very common. This is an unusual type, okay, found in the order Conjugales. Okay, in Chlorophyceae, in green algae, in the other Conjugales, this type of sexual reproduction is usually found. Here, what happened? The Amoeboid Gametes, okay, the Amoeboid Gametes, they will, also known as a Plano Gametes, they will fuse to each other. As you can see, this is the female and this is the male. Alright, this is the male, this is the female. As you can see here, it is written, they will just fuse with each other through this tube. Okay, there will be a tube formation known as conjugation tube, allowing the amoeboid movement of these two spores. They will come in contact with each other, they will fuse, and zygospores are formed. Okay, so this can be seen in case of green algae such as Zycnema and in Spirogyra. So these are the different types of reproduction in green algae, that is Chlorophyceae. Now we also have this one type here, that is parthenogenesis. Now what happened in parthenogenesis? Sometimes the perfect spores, they are attained without fusion. Okay, and the male and the female, they will not fuse together. So those spores are referred to as azygospores or parthenospores. Here, the female gamete, it may give rise to a new plant without fusing with the male gamete, and the process is termed as parthenogenesis. Okay, remember this one, that here the female does not fuse with male gamete, but it develop into a new plant through a process known as parthenogenesis. And this process, it has been recorded from again from several Chlorophyceae. Now sometimes one species of the genus, it reproduces parthenogenetically and the other species does not. For example, Ulva lactuca is parthenogenetic species while Ulva lobata is not. So that means in the same genus, okay, we know that we have many species, right, in a single genus. For example, Chara, also we have many, Erugonium, Ectocarpus, we have many species. So you can see the example here, the two different species of Ulva, of the genus Ulva, that is species Lactuca and Lobata. These two species, in these two species, the sexual reproduction is different. As you can see in Ulva lactuca, the sexual reproduction, it occurs through parthenogenesis, but this is not the case in Ulva lobata.